Okay, so the first thing we need to do is understand what the P generation was. And his cross for P generation was he did purebred tall, and he crossed it with a purebred, or truebred would be the same as well, short. And that just means that if that plant were to self-pollinate itself, it would be, short. If it were to self-pollinate itself, it would only produce offspring that look like itself. So this would only produce tall. So that means that both of them are tall. Both of the alleles are tall. And both of the alleles are short. And so let's look at this and understand the difference between a genotype and a phenotype. Right? Big T, that is a genotype. Right? Those letters, the words that describe those two letters is homozygous, dominant. Now, if you had one of each, you have hetero, which means different, and homo means the same, referring to the same allele in both of the gametes that were combined to form the zygote. Heterozygous, means that the zygote that was formed by the combination of sperm and egg, or pollen and egg, had two different alleles. There are two different forms of homozygosity. You could be homozygous dominant, or you could be homozygous recessive. I had students ask me before, I'm reading a word problem and it said cross two heterozygotes. How do I know if it's heterozygous dominant or recessive? And the answer is, you don't. It doesn't matter. If it says it's heterozygous, that means it's just one of each. So let's look at this and understand that these are the descriptions of the letters, right? Now, if we look at the phenotype, right? The phenotype, there's only two phenotypes, right? Tall and short. And the genotypes that match, well, the two genotypes that will give you tall are big T, and big T, little t. The two genotypes that will give you short are just one genotype will give you short. So if it is a short, you know it's homozygous right off the bat. So let's go back to that P generation. And let's look at his original cross. Your guys' Punnett Square is a way to show the possible offspring from this cross. We put the parents' gametes at the top. This represents the parent's allele. And there is only one allele, one version of that gene in each gamete. So when this parent right here goes through meiosis and separates their chromosomes into gametes, sex cells, half of them will have this allele and the other half will have this allele. Now I'm going to put this parent on the side. It doesn't matter which parent I put on the top and which parent I put on the side of the Punnett Square. When we combine them, this allele goes here and this allele goes here, right? This allele goes here and this allele goes here. So we get a big T, little t, big T, little t. Okay? Same thing here, big T, little t, big T, little t. And so what's inside of the Punnett Square is the offsprings. Right? Genotype. Or the possible, or the probable, the possible offspring genotype. Okay? So that is the first generation. So the offspring, the children of the P generation are the F1 generation. So now I want to cross two F1s. Okay? Now Mendel planted hundreds of seeds that were formed between this cross. And he just took two plants and he crossed them. So he wanted to cross two F1s. So let's look at what that F1 cross looked like. He took two hybrids. A hybrid is a cross between two purebreds. And so each one of these is a hybrid. That's a hybrid and that's a hybrid. And now we're going to set up the Punnett Square. Now you have to remember, guys, that the law of segregation just describes what happens when we create our sex cells. Now these plants have to then create their sex cells. Half of the pollen will have the big T, and half of the pollen will have the little t. But the same goes not just with the pollen cells, but also with the eggs. Half of that plant will produce eggs that have a big T, and half will produce eggs that have a little t. If you look at a flower, it has the pistil, right? That's where the eggs are produced in the middle. And then it has these stamen, right, that have all the pollen in it. So these cells, when they go through meiosis, right, meiosis is happening here and it's happening down here, right? These are the pollen up here, right? There are the ones that are going to be either big T or little t. And this is the egg right here. That's going to be either big T or little t. Now remember, this is a plant, right? And then it has all the flowers right here. Like my flowers? Okay? So either a big T pollen is going to go down here and fertilize this, or a little t. It's either going to fertilize a big T egg or a little t egg. So if this pollen fertilizes this egg, you're going to get a seed or a zygote, which will be planted and grow into a plant that has the genotype of big T, big T. But if that pollen happened to fertilize that egg, then you would have gotten a big T, little t. And by the way, it's not only just this plant self-fertilizing itself, it's this plant fertilizing other plants as well. If this pollen were to fertilize that egg, right, then you're going to have a big T, little t as well. And only if this pollen fertilizes this egg would you get a little t, little t. And there is equal chance, 1/4, 1/4, 1/4, 1/4, of that happening. Okay? And so when you do the Punnett Square to represent that, right, this guy who produces these possible gametes is going to cross with this plant. It's not really a guy or girl, they're plants, right? So I don't know why I keep on calling them that. You're going to look at a Punnett Square that looks like this. So I'm going to put this parent on the top of the Punnett Square and the other parent on the side. It doesn't really matter. And then I combine. Now let's look at the genotypic ratios. Okay, guys, what genotypes do you see in the children? These are the children, the F, what? 2s. Because these guys, right, up here are the parents, the F1s. And so if you look at the genotypes, look, I have a big T, big T. How many of the 4? Well, it looks like 1 out of the 4 possible combinations are homozygous dominant. 25%. What else do I got? Oh, looks like I have these guys right here. Right? Those guys are 2 out of the 4. That's 50%, right? And then I got this guy right here. That's 1/4. That's 25%. So the ratio is a 1 to 2 to 1 ratio. For every one of these, I have 2 of those and 1 of those. Now let's look at the phenotypic ratio. It's a little bit more interesting. Let me clean this up a little bit. Well, this guy's going to be what, class? He's tall. That's his phenotype. This guy's phenotype is? What? Tall. Because the dominant one masks the recessive one. This one will be? And this one is? Short. So the phenotypes, these 3, right? We have tall. Well, we have 3 out of the 4 are tall. That is a 75%. How did I calculate that? Well, 3 divided by 4 equals 0.75. You multiply that by 100 or just go bam, bam on the decimal point, and that's going to give you 75%. And so 1 divided by 4 is 0.25, which you just got to move 1, 2 to the right, and that's going to give you 25%. Okay? And so the short is 1 out of 4, as you can see right here. That's 25%. So now let me throw a little curveball at you. Okay? Slow down. You'll be all right. Everybody, we'll all make it together. What if we have... Well, let me use T's. We'll get the H's in our lab. Let's just stick with the same letters. What if we have a tall plant, right? That's heterozygous. This is tall. And we cross it with a short plant. So let's all do that Punnett Square either in our head or in that blank piece of paper that we did our warm-up on. And we'll see that I just chose to put this parent on the top. I could have easily put it on the side. It doesn't matter. I put the other parent on the side. Now these are the parental alleles, or the gametes, right? This would have been like, oh, what if this sperm fertilizes this egg? And so in the middle you have this. Now obviously these are plants. They don't have sperm. This is combined. Okay, so let's look at the genotypic ratios. So it looks like we have, these two are big T, little t, right? 2 out of 4, 50%. And then we have a different genotype, not the same. Homozygous recessive, also 2 out of 4, 50%. Okay, so that's a 2 to 2 ratio, which can be simplified as 1 to 1. All right, but now same cross, nothing's different. Tall, short, tall, short. So now I need to look at the phenotypic ratios, right? Ratios. That's what happens when you write faster than you think. What are the different types of phenotypes? Well, I have tall and I have short. That's what I have. I have tall and I have 2 out of 4, which is 50%. And I have short. 2 out of 4, 50%. Okay, last curveball, I promise. What if, okay, I have a, oops, why is that blue? We'll stick with, you know what? I want to stick with the theme. Hold on. Save your questions till the very end. Now I got homozygous. This is the tall with the tall, but they're different genotypes. Before I had the exact same genotypic ratio as phenotypic ratio, right? So genotypic ratios. Is this the exact same cross as I had before? No, it wasn't. The cross that I had before, was it not this? Wasn't it that? That's what I had before. I had a heterozygote with a homozygous recessive. Now I have a homozygous dominant with the heterozygous. Not the same thing. So let's look. Before I had the same 50-50 with the genotype as I did with the phenotype. Looks like I got 2 out of 4, or 50%. It's looking eerily similar to the last problem. Right? But now, let's look at the phenotype. Looks like this is going to be a... Why am I using H's? I just switched. Nobody noticed that I was using H's? Keep your questions at the end. Those should all be T's, but whatever. You guys are smart enough to figure that out. Hold on. Shh. I'm screencasting, so be quiet. So these are all tall. Now, there's only one tall. 4 out of 4, 100%. Okay? all of these should be T's, right? So you should, if you guys really want to have this correct, all of this should go back. If you want to really stay consistent, right, it should all be like this. Okay. Right? And then the stuff on the inside, right? Just so we're not confused. Is that.